And what I'm going to tell you is, how does this work? About molecular stratification of breast cancer and how I think it's reaching the clinic. And instead of asking, can we afford to do it, I would put the question the other way around. Can we continue not to do it? Is that the way to continue to practice oncology? And I think when you hear a lot about stratified medicine, frequently people are thinking of tumors like melanoma or glioblastomas, like you've just heard, where really the prospects are dismal. In breast cancer, the survival at 10 years of breast cancer treated in our unit is 80%. So it's a completely different clinical scenario. And I want you to keep that in mind. It's a very frequent disease. There are 45,000 cases diagnosed every year in the UK. And there are still about 12 to 15,000 women dying of breast cancer. So it's a public health problem. Now, when I finished medical school, more than 25 years ago, I haste to say, the survival of breast cancer was 50%. So what has made that 30% difference? 30 out of every 100 women are now surviving breast cancer that were dying 25 years ago. It's a mixture of things. It's screening programs, adjuvant hormone therapy, the most maligned but best cancer drug ever discovered by us, tamoxifen, very effective drug, very cheap. Adjuvant chemotherapy, of course, trastuzumab for the 12 to 15% of women that are HER2 positive, not 30% like a lot of leaders keep saying, is 12 to 15% if you look on a population basis. But I told you that 50% were cured when I finished medical school. Now it's 80. What is the price that we pay for that, besides the price of treatments? Is that we grossly overtreating a large number of women. And so in breast cancer, molecular stratification is absolutely essential, and we're failing women if we don't do it. We want to slice the ones that we are overtreating, and there is a large number that we are overtreating. And we want to identify very early on the ones that we are failing, 20 out of every 100, to direct treatments to them. So today, we have two markers that, if well done, I have not seen any evidence that there is any gene expression signature that outperforms these markers, and that's ER and HER2. And just with these two markers, properly done, you can separate breast cancer into 4 diseases. ER positive, HER2 negative disease, I'll come back to that one, which is the most common. Then that's 70% of breast cancers. 15% of breast cancers will be HER2 positive. They are distributed equally between ER positive and ER negative. And for this, we have a predictive marker that we have to do well. There is no gene expression signature that is better than doing the immunohistochemistry in the fish, and we now have treatment for them. It doesn't work for everyone, but we do. And then tumors that are ER negative and HER2 negative, they are about 15%. When you look, they are also PR negative. They're called triple negative. I'm not going to spend any more time on this very fashionable class. And one of the reasons is that I would have to spend a whole lecture on it because it is not a uniform entity. Again, contrary to what a lot of people in the community keep insisting on or persisting on wrongly saying that it is an entity, because it's not. And it is not equivalent with basalness, and it's not equivalent with brachythness. Now, to this group, this is the group where we need to do better, because that's the group where a lot of women are cured with hormone therapy alone. A small percentage benefit from currently available chemotherapies at the cost of a lot of toxicity, but it saves their lives. But a large number are receiving unnecessary chemotherapy, for which they get all the side effects, including secondary leukemias, heart trouble later in life, etc., without any benefit. So it is for these that we need to do better. And actually, we know that all of the oncotype DXs and the GenDias, etc., what they do is to really rate these in terms of proliferation. And there are proliferation markers that you can use. And actually, they have been extensively validated, extensively, to a degree that neither or none of the gene expression signatures has. We have validated BCL2 in 11,000 breast cancers from 5 different countries, many different hospitals. And a breast cancer that expresses a lot of BCL2, whether it's in Europe, in the United States, in Australia, it's a better prognosis breast cancer. We can debate as how you standardize doing Ki67, but it can be standardized. And if you just combine the two, and this is work done in collaboration between my group and group in Vancouver, and just to give you an idea, this is data from 3,000 cases, and then externally validated in a further 4,000 cases from Vancouver. Just doing a simple index of Ki67 and BCL2, you can separate dramatically the ER positive cases into cases that proliferate a lot, proliferate very little, and somewhere in between. And that is prognostic. But none of this tells us anything about the biology of the disease. Now, is Ki67 the best proliferation marker? Probably not. This is just work from about 3,500 cases. We have not yet externally validated it. We looked at 6 proliferation markers, some combinations between them to try to classify cells to the cell cycle stage. It looks like our aurora kinase A by immunohistochemistry is better than any other of the markers in a multivariate model. So this is the state of play. ER, PR, and the proliferation marker in combination with BCL2. And I think if you do this properly, there is no gene expression signature that outperforms this. And do we really care, or do we really want to know, or do we want to get at the biology of the disease and at treating different cancers differently? Now, if you look with one classifier, the intrinsic classifier, that's basal-like, that's the HER2 subtype, luminal A's, luminal B's, and normal-like. And what I have here is a heat map of copy number aberrations, somatic copy number aberrations in the tumors. And you look at any of your favorite subtype, and you see that they are extremely heterogeneous as far as the genomic composition of the tumors. And we know that cancers arise by the accumulation of mutations, including copy number gains and losses. So we decided to, if you want, settle the issue. And this is the paper that came out a couple of weeks ago in Nature, where we looked at 2,000 tumors using a combination of array CGA gene expression profiling to really show that the disease is divided into 10 subgroups. And these 10 subgroups are exquisitely characterized by copy number aberrations that contain oncogenes and tumor suppressor genes that classify the breast cancer into 10 different diseases. And here you have a heat map that summarizes for each of the 1,000 loci that we looked at to build this classification, summarizes both the copy number state and the gene expression state. And red, bands of red represent copy number gain associated with overexpression, and blue the opposite. And you see that, you know, they fall neatly into 10 very robustly different diseases. So you have, for example, cluster 1, these are ER positive tumors. They are some of the luminal Bs, and they have amplification distal to HER2 on chromosome 17, and an amplification of 20q. For the sake of time, I'm just going to not dwell a lot on these subtypes, but there are 10 different groups. And I talked about that yesterday in my lecture. Now, if you want any reassurance that these 10 groups are pretty robust, this is the data that should reassure you. Because these are the first 1,000 tumors where we derived the classifier. These are the second 1,000 tumors. And the top bars on charts on each of these 10 groups is a summary plot of the copy number changes. And above the line are copy number gains and amplifications represented in red. Below the line are copy number deletions and homozygous deletions represented in blue. And whatever class you look at, whether you belong to the first 1,000 tumors or to the second 1,000, they are pretty much nearly identical, showing that these are very robust classes indeed. We did many statistical things to show that this is the most parsimonious number. Could it be 9, 12, 13, or 10? 10 is better than any other of the combinations that we looked at. Now, importantly, do these classes carry more prognostic information than the intrinsic classification done in exactly the same tumors? And the answer is absolutely yes. So these are 1,000 tumors, luminal A, luminal B, normal-like, HER2 subtype, basal-like, exactly like people have described. Annelies in the back was the pioneer of this classification. It is very prognostic, with a very highly statistically predictive p-value. But when you look at the classification that we now produce, it's even more discriminatory with a much higher p-value. And I'm just going to focus on one class. So by the way, you might wonder, what are these horrible breast cancers here? This is HER2 positive disease prior to trastuzumab. None of these women received trastuzumab in the adjuvant setting because their tumors preceded the seminal ASCO meeting where the findings of the adjuvant trials were reported. Those tumors there that are horrible in the first 5 years and then, like high-grade lymphomas, appear to be cured because the curve flattens, are basal-like breast cancers. Now look at this curve here. If you track it back, these tumors are all ER positive. In the first 5 years, they have significantly worse prognosis than the majority of other ER positive disease. They have significantly better prognosis than the worst forms of breast cancer. But after 5 years, these women tragically continue to die of breast cancer. These are about 5% of breast cancers. These are highly aggressive ER positive breast cancers that have 2 cassettes of amplification on chromosome 11 that we are failing today with hormone therapy, and I think these are the ones that will require adjuvant chemotherapy. Well, that is going to be, it's to be debated. Whether we can find targeted therapies for this subgroup, I believe we can. Now, just to illustrate how, if you look at the biological classification in a very large number that tell you something about pathogenesis, because I didn't have time to go into the details, but different networks of oncogenes and tumor suppressor genes will be involved in the different subtypes. These are all tumors that are ER positive and PR negative from our series, and I'm about to finish. So these are about 1,700 tumors, so this is as robust as it gets. So we're now looking at adding the proliferation marker. And if you are proliferating more by Ki67 here, measured by gene expression, you do much worse and you clearly separate. I apologize, the scales are different because we did the analysis using 2 different programs, but you know, the results are absolutely, I need to make a new slide. Look at the 7 classes of ER positive, and these are the horrible ones I told you before. These are excellent prognosis ones, and then intermediate ones. And of course, now we overlay onto this mutational analysis, but it's by 10-fold order of magnitude different. So this is a paper that myself and Annelies contributed tumors to, but the analysis was led by Mike Stratton. This is the pattern of mutations in ER positive, in ER negative disease. You see that a lot of these aberrations are copy number aberrations, and you have point mutations. There are tumors, ER positive tumors with lots of mutations, tumors with very few, same in ER negative. There are genes that are very commonly mutated or copy number amplified or deleted, and then you have a long tail of less frequent genes. And this is the other paper that also looked at 100 tumors, again, a mix of ER positive and ER negative, and came up with some new genes also. So, in conclusion, we will divide breast cancer into at least 10 subtypes using an array, maybe PCR, but I would say that what we're going to use is going to be sequencing based, because with very low coverage of sequence, you can get the same type of copy number information that we get today using arrays, and that is being shown by many different groups. I think that we will, for each subtype, sequence not a scary number of genes, but somewhere between 25 and 75 driver genes. That is the best guess that we have at the moment. And we will sequence these deeply, because there is clonal heterogeneity in tumors, and there are subclones that, as we've learned in colorectal cancer, Jose Tabernero is there, he wrote the news and views on 2 seminal papers in Nature last week, showing that minor clones of KRAS mutant cells exist in colorectal cancers that then emerge and are responsible for resistance to cetuximab. So you'll have to deeply sequence these drivers, maybe 500-fold, maybe 1,000-fold. This will not cost an inordinate amount of money. And we will use the above information to decide treatment and to monitor treatment by using barcodes that each of these tumors will produce, and we can monitor them on circulating tumor DNA. I don't have time to go into that. Please look at poster 876 from one of the postdocs in the institute that has done the work that illustrates the value of this. Thank you.